Hello everyone, welcome into the ITLS Academy. Myself, Dr. Himani. I'm your mentor in this animal biotechnology course. So today our lecture on polymerase chain reaction. So what is PCR actually? The polymerase chain reaction is a common technique practiced in genetic laboratories because it is a basic requirement for a genetic or molecular lab. Replication is a process of DNA synthesis. However, for us, mimicking replication in a lab is not possible. But after the discovery of the thermostable DNA polymerase, the dream of synthesizing DNA in a lab has come true. The polymerase chain reaction is one of the emerging scientific techniques and has infinite opportunity in research as well as diagnostics. Different variation in the native PCR helps in the development of different techniques for different application. So we can say like that PCR is an amazing process in which a specific region of DNA is replicated over and over again to yield many copies of a particular sequence. The most widely used target nucleic acid amplification method is the polymerase chain reaction. This method is able to amplify a single copy of a nucleic acid target, often understandable by standard hybridization method, and multiply to 10 to the power 7 or more copies in a relatively short period. So, how we can define it? PCR is the polymerase chain reaction is techniques used in molecular biology to amplify a single copy or a few copies of a segment of DNA across several orders of magnitude genetic thousands to millions of copies of a particular DNA sequence, or we can say PCR, a polymerase chain reaction, is a cyclic temperature dependent reaction used to amplify the gene of interest. So let's discuss about the history of the PCR. In 1983, Kary described the techniques of in vitro gene amplification and named it as a polymerase chain reaction. So later on he was awarded the Nobel Prize for his finding. However, the story of PCR was begun when the DNA polymerase was isolated from the thermostable bacteria. In 1996, Thomas had discovered the bacteria from the hot spring of water and named it Thermus aquaticus. Later on, in 1976, Chain et al. Isolated DNA polymerase from Thermus aquaticus, named it as Taq DNA polymerase. The overall idea of the polymerase chain reaction is to get copy of DNA or gene we wish to study. We cannot visualize a few DNAs. That is why we need to amplify DNA. After the isolation of thermostable Taq DNA polymerase, the idea of temperature dependent amplification came in the picture. The PCR chain reaction is temperature dependent process of DNA amplification. The machine used in the PCR technique is known as a thermocycler. So now discuss about this thermocycler machine. This thermocycler, also known as the thermocycler PCR machine or DNA amplifier, so is a laboratory apparatus most commonly used to amplify segment of DNA via the polymerase chain reaction. The cycler then raised and lowered the temperature of the block is discrete pre-programmed step. The machine thermocycler provides various temperature for each step to complete. You can see here different variation temperature for each step to complete it. There is a different variation. It's a temperature is 72 degree, and when the template DNA is degraded, then temperature is 94 degree, and again the next process is called annealing. Here the temperature is 55 degree centigrade. So you can say like that this machine have the various temperature for each step to complete. Denaturation, annealing, this is the denaturation, and denaturation, annealing and extension of DNA occur at different temperature. Thus, the machine is known as thermocycler. The overview of every PCR cycle at various temperature is given below. You can see it here in this picture. So like you can see in this picture, like just the denaturation, in the denaturation process the temperature was 94 degree, and in the annealing process temperature was the 55 degree, and if we are seeing here in this step the extension process temperature is 72 degree. So this machine have a different temperature for each step to complete. So let's discuss about the requirements used in the PCR. First one is the chemicals. DNTPs, distilled water, PCR reaction buffer, enzyme tech DNA polymerase, primer, and templates DNA are used as a chemical. And if we are talking about the instruments, then thermocycler, spinner, and agrogel electrophoresis unit used as instruments, and other utilizes is PCR tubes, stands, pipettes, and tips. So common components of PCR. Common components of PCR constitute the following. First one is DNA templates. The double stranded DNA of interest separate from the sample. Next one is DNA polymerase. Usually a thermostable tech polymerase that does not rapidly denature at high temperature. It could be 98 degree and then can function at the temperature optimum of about 70 degree. Next is the oligonucleotide primer. Short pieces of single stranded DNA, often 20 to 30 base pair, which are complementary to the three end of the sense and antisense strand of the target sequence. Next is the deoxynucleotide triphosphatase. Single unit of the base adenosine, tyrosine, guanosine, and cytosine. You can see DATP, DTTP, DGTP, and DCTP provide the energy for polymerization and building block for DNA synthesis. And next one is the buffer system. Includes magnesium and potassium to provide the optimal conditions for DNA denaturation, and renaturation also important for polymerase activity, stability, and fidelity. So here we are going to understand about the principle of PCR. Here in this picture you are seeing three steps: cycle 1, cycle 2, and cycle 3. Let's discuss about the first is cycle 1. So the target sequence of nucleic acid is denatured to single strands. You can see here is the double strands of DNA. So the target sequence of nucleic acid is denatured to single strand. Primer specific for each targeted strands. Again I am repeating, primer specific for each targeted strands. Sequence are added and DNA polymerase catalyze the addition of deoxynucleotides to extend and produce new strands complementary to each of the target sequence strands. This called the cycle 1 process. And if we are talking about the second cycle, then in cycle 2, both double strands, these both double strands produced of cycle 1 are denatured and subsequently serve as target for more primer annealing. You can see here, for more primer annealing and extension by DNA polymerase. This is the process of cycle 2. And if we are talking about the cycle 3, then after 25 to 30 cycle, at least 10 to power 7 copies of targeted DNA. These are the targeted DNA may be produced by means of this thermal cycling. You can see here, these are the cycle 3. So, what is the requirement for PCR? A PCR reaction contains the target double standards DNA, two primers that hybridize to flanking sequence on opposite strands of the target. All 4 deoxyribonucleoside triphosphatase and DNA polymerase along with buffer cofactors of enzymes and waters. Since the reaction predictably become heated to high temperature, PCR depends upon using a heat-stable DNA polymerase. Many such heat-stable enzymes from thermophilic bacteria are now available commercially. The first one and the most commonly used in the Tec polymerase from the thermophilic bacterium Thermus aquaticus. So next is the step involved in the PCR. This is the very important step. So please concentrate on this slide. The step involved in the PCR is 3 type of step involved in the PCR. First one is extraction and denaturation of target nucleic acid. Second one is the step in amplification. In this process, 3 steps are used. First one is denaturation, second one is primer annealing, third one is extension. And last step involved in the PCR is product analysis. So this you can see here. First we will discuss about the extension and denaturation of target nucleic acid. Then we will discuss about the step in amplification. In this process we will discuss denaturation, primer annealing, extension. And last we will discuss product analysis. So first one is the extraction and denaturation of target nucleic acid. For PCR, nucleic acid is first extracted from the organism or clinical sample potentially contain the target organism by heat chemical or enzymatic method. You can see here, these are the nucleic acids. Here is the DNA sample you can collect by the targeted organism. So one extracted target nucleic acid is added to the reaction mixture containing all necessary components of for PCR. It could be the primers. You can see in this here, the all necessary components could be the primers, nucleotides, and covalent ions, buffers, and enzyme. Here we will use tech polymerase and place into a thermal cycler into undergoes amplification. So in the first step, extraction and denaturation of target nucleic acid, we just prepared a sample solution for the amplification by the thermal cycler. Next we will discuss step in amplification. So first is, we can say conventional PCR involve 25 to 50 repeated cycles, which each cycle comprises 3 sequential reactions. First one is the denaturation. So the reaction mixture is heated to 94 to 95 degree for a short time period, about 50 to 30 seconds, to denature the targeted DNA into single strands. You can see in this picture, first step is denaturation. So here, this is our sample. So the reaction mixture is heated to 94 to 95 degrees centigrade for a short time period, about 50 to 30 seconds it could be. So 50 to 30 seconds, or to denature the target DNA into single strands that can act as template for DNA synthesis. You can see here, these are the single strands after the denaturation, and these single strands act as a template for DNA synthesis. So again I am repeating, first step is denaturation. The reaction mixture is heated to 94 to 95 degree for a short time period. The short time period could be 50 to 30 seconds, to denature the targeted DNA into single strands that can act as template for DNA synthesis. Next, we will discuss about the primer annealing. The mixture is rapidly cooled to the defined temperature, which allow the two primers to bind to the sequence on each of the two strands flanking the target DNA. And in this step, you can see here, the mixture is rapidly cooled to the defined temperature. Temperature is 54 degrees centigrade. And this temperature allow the two primers. Here is the primers. You can see here is the primers. Here the temperature allow the two primers to bind to sequence on each of the two strands flanking the targeted DNA. Primers are short. You can see in this picture, primers are short, not the long type of primers we are using here. So primer are short, single stranded sequence of nucleic acid. It could be the oligonucleotide, usually 20 to 30 nucleoside long, selected to specifically hybridize to a particular nucleic acid target, essentially functioning like probes. So this annealing temperature is calculated carefully to ensure that the primer bind only. These primer bind only to the desired DNA sequence. Usually this temperature use 54 to 55 degree. So this annealing temperature is calculated carefully. Again I am repeating, this annealing temperature is calculated carefully to ensure that the primers bind only. These primers bind only to the desired DNA sequence. One primer bind to each strands. One primer bind to each strands. The two parental strands do not re-anneal with each other because the primer R is large excess over parental DNA. Next we will discuss about the third process is extension. This is the extension process. The temperature of the mixture is raised to 72 degree usually. In this extension temperature is 72 degree and kept at the temperature for a preset period of time to allow DNA polymerase to elongate each primer by copying the single standard templates. Annealing of primer to target sequence provide the necessary template format. So again I am repeating, annealing of primers to target sequence provide the necessary template format that allow the DNA polymerase to add nucleotide to the 3' terminal end of each primer. 3, you can see here. These primers bind to the 3' terminals end of each primer and extend sequence complementary to the target template. Taq polymerase is the enzyme commonly used for primer extension. Taq polymerase is the enzyme commonly used for primer extension, which occurs at 72 degree. The enzyme is used because of its ability to function efficiently at elevated temperatures and to withstand the denatured temperature of 94 degree through several cycles. The ability to allow primer annealing and extension to occur at elevated temperature without determining to the polymerase increase the strength of the reaction, thus decreasing the chance of amplification of non-target nucleic acid, or we can say is a non-specific amplification. So here we already covered these 3 steps. These 3 steps was very important for the amplification of sample. First one was the denaturation, and second one was the annealing, and third one was the extension. You can see also in this picture why double standard DNA in first step denaturation stage temperature was the 94 to 95 degree, convert into the single strands, and again the annealing strands, this primer bind with this single standard DNA, temperature was the 50 to 56 degree, and in the third step the extending stage temperature was the 72 degree. Here the template are bind with the targeted DNAs. So the 3 steps of the PCR cycle are repeated. Thus in the second cycle the 4 standard denature bind primer and are extended. No other reactants need to be added. The 3 steps are repeated for a third cycle and so on for a set of additional cycles. By the third cycle, some of the PCR products represent DNA sequence only between the 2 primer sites and the sequence does not extend beyond these sites. As more and more reactions cycle are carried out, the double standard DNA are synthesized more in numbers. After 20 cycle, the original DNA has been amplified a millionfold, and this rise to a billionfold million after 30 cycles. So next we will discuss about the using gel electrophoresis to visualize the result of PCR. The result of a PCR reaction are usually visualized using gel electrophoresis. Gel electrophoresis are techniques in which fragments of DNA are pulled through a gel matrix by an electronic current, and it separates DNA fragments according to size. A strands of DNA ladder is typically included so that the size of the fragments in the PCR sample can be determined. DNA fragment of the sample length from a band on the gel, which can be seen by eye if the gel is stained with a DNA binding dye. You can see the band of the you can see a bend in the gel because this gel is stained with the DNA binding dye. For example, a PCR reaction produce 100 base pair fragment. You can see here these are the PCR reaction. These are the DNA ladder. DNA ladder have different type of base pair is here, but when PCR reaction produce 100 base pair fragments, 400 base pair fragments, and then it would look like this on a gel. You can see this is a 100 base pair band. So by the DNA ladder, we can identify this band could be the near about the 100 base pair. So a DNA band contains many copies of the targeted DNA region, not just one or few copies, because DNA is microscopic. Lots of copies of it must be present before we can see it by eye. This is a big part of why PCR is an important tool. It produces enough copies of DNA sequence that we can see or manipulate that region of DNA. Now we will discuss about the application of PCR. PCR can amplify a single DNA molecule from a complex mixture, largely avoiding the need to use DNA cloning to prepare that molecule. Variant of the techniques can similarly amplify a specific single RNA molecule from a complex mixture. DNA sequence has been greatly simplified using PCR, and this amplification is now common. Using PCR, a DNA sequence can be amplified million or billion of times, producing enough DNA copies to be analyzed using other techniques. By using suitable primers, it is possible to use PCR to create point mutations, detections, and insertions of the target DNA, which greatly facilitated the analysis of gene expression and function. PCR is sensitive and can amplify vanishingly small amount of DNA. Thus, using appropriate primer, very small amount of specified bacteria and virus can be detected in tissue, making PCR invaluable for medical diagnosis. PCR is used in many research labs, and it also has a practical application in forensic genetic testing and diagnosis. Because of its extreme sensitivity, PCR is now fundamentally important to forensic medicine. It is even possible to use PCR to amplify the DNA from a single human hair or microscopic drop of blood left at the scene of a crime to allow detailed characterization. PCR can also be used to test for a bacterium or DNA virus in a patient's body. If the pathogen is present, it may be possible to amplify regions of its DNA from a blood or tissue sample. So guys, I have covered my topics related to this lecture. So I hope you all students able to understand this lecture. If you have any query, then you can mail me. So thank you for listening me. See you in the next lecture.